And we've just received a message that Professor Aikawa is joining us online from Boston, I guess. So I don't know whether we can get the connection and whether we can welcome him among us thanks to the miracles of internet. And we can. So Professor Aikawa, can you hear us well? We can see you nicely, and we are very much looking forward to your contribution to our program. Hello, hello, and welcome.
Good morning. Thank you very much for this great opportunity to share with you, what we have found, in terms of the effects of Pemafibrate. Particularly today, I'm focusing on its effects on inflammation in vein grafts, AV fistulas, and post-stent coronary arteries. I'm going to share with you mainly these two studies that we published in Circulation 2021 and JHA 2022 on the effects of Pemafibrate on vein grafts and also in-stent restenosis in coronary arteries in pigs. Okay. So in this schematic, I'm demonstrating that the activated macrophages promotes vascular diseases, including atherosclerosis and the vein graft disease and peripheral artery disease, no matter where those pro-inflammatory phagocytes come from. When I was working with Peter Libby in Brigham and Women's Hospital, and we demonstrated that lipid lowering by either diet or statins can reduce macrophage accumulation and activation. And that's our typical features of atherosclerotic lesion prone to acute thrombotic events in these publications, which provided the molecular basis for so-called plaque stabilization hypothesis. The very good news is, our findings were consistent with the many statin mega trials, including this Jupiter, led by our colleague Paul Ritker in Brigham Cardiology, that demonstrated that cholesterol lowering prevents acute coronary events. However, as many of you know, that statins reduce cardiovascular events in many, but not all people. So therefore, our mission in the past couple of decades, has been to identify new mechanisms for macrophage accumulation as therapeutic targets. However, we also recognize that there are many major challenges in drug discovery. First of all, discovering promising targets takes long time, and also success rates of new drug targets in clinical development are extremely low. So why there are such problems? The one reason might be the human disease mechanisms are very complex, much more complex than we traditionally thought. And then, such a linear hypothesis is not really, modeling the real human disease mechanisms. Actually, disease mechanism of each, for example, cardiovascular disorders, is buried in the middle, very complex, network of biological, pathological pathways. We therefore decided to shift our focus from hypothesis-driven research to systems approach, over a decade ago. Another challenge is that there is a huge gap between academia and industry. Many good discoveries that we make in academia cannot really cross this huge valley of death. And many good ideas die without reaching the stage where you can actually develop the drugs, with industry partners. So therefore, we realized that we would need to establish new paradigm of drug development, something like this. In collaboration with COA, we established the new Center for Interdisciplinary Cardiovascular Sciences, within Brigham Cardiology, in 2009. So these are our members from several years ago, and we have nice facility thanks to support from COA. In order to establish systems approach, we have quickly implemented the innovative technologies such as proteomics, systems biology, our own bioinformatics programs, and computational drug screening system, and recently AI. Then we started working on this paradigm several years before that NHLBI emphasized that systems biology is needed to identify innovative drugs. So our center is obviously between the collaboration with academia and industry, and also between Japan and US. But more importantly, we are a very dynamic interface of medical science and data science. Typically, we start each project, with omics screening, and the data processed with bioinformatics, including AI. And then very importantly, we use network analysis powered prediction to really assess the potential impact of your target in the future clinical trials to increase the success rates. Then we, of course, do comprehensive in vivo, in vitro, mechanistic studies before, drug screening begins. As I mentioned, that our concept is to merge, academic target discovery research and drug development in industry. So this kind of bridge is quite short as compared to traditional target discovery efforts without close connection between academia and industry. So I'm going to talk about the vein graft failure today. And as many of you know, that due to diabetes pandemic, this problem has become really, really serious. And the increasing prevalence of PAD, makes this disorder, the global, health burden. The problem here is 30 to 50% of PAD vein grafts narrow or are occluded within 1 year. Then, despite this clinical impact, current options include revascularization or amputation. The problem here is that we have no medical therapies available. And however, if you look on the bright side in terms of drug development, high initial year event rates makes it possible for us to run small and short clinical trials. So this is quite attractive target for new drugs. We are interested in a macrophage biology, as I mentioned earlier today, and then accumulating evidence suggests that plaque rupture occurs in even vein grafts that are rich in macrophages. Then we formed the interdisciplinary a research team of scientists and to conduct systems biology powered target discovery research against vein grafts and AV fistula disease. The first paper published in Circulation this paper focused on the effects of Pemafibrate. Okay. We use the mouse model of vein graft disease, where we transfer the IVC to the carotid artery in LDA receptor knockout mice. In this model, you can see the development of very complex vein graft disease, which is almost similar to complex coronary artery disease within a month or so. So using this model, we performed unbiased proteomics analysis. Left side, I'm showing the static one-time point tissue layer proteomics. We also performed the proteomics of vein graft and control IVC over several time points. As you can see here, that okay, yeah. While IVC, proteome doesn't really show so many changes over time. In vein grafts, you can see the expression patterns of proteins, keep changing, over time. So from this, large data set, we analyze the data, in multiple different ways. In this, comparison, between the lesion and control IVC, we found that PPA alpha is suppressed during development of vein graft lesions. Then, we got very interested in this data. And our question was, does PPA alpha have a causal role? Okay, so vein graft, a failure is a global health burden, as many of you know, due to diabetes pandemic that causes increased prevalence of PAD. The problem is that 30–50% of PAD vein graft narrow or occluded within 1 year. And despite this clinical impact, current options include revascularization or amputation. Then, unfortunately, we have no medical therapies available. But if you look on the right side of this serious disease, due to high initial year event rates, the clinical trials for a new drug against this disease could be small and short. And so therefore, we are particularly interested in this disease context. Next slide, please. Okay. Accumulating evidence demonstrated that plaque rupture occurs in human vein grafts, like plaque rupture in coronary arteries, and then many human vein grafts are rich in macrophages. So therefore, we are targeting inflammation in vein graft disease in the mice center. Please, next slide, please. Okay, we formed a cross-disciplinary research team of investigators to conduct systems biology target discovery effort against vein graft disease and AV fistula disease. Next slide, please. So first paper from this effort published in Circulation in 2021, focusing on the effects of Pemafibrate on vein graft disease. Next slide, please. So we use mouse model of vein graft disease, where we transfer the IVC to the carotid artery in LDA receptor knockout mice. In this model, so you can see the quite advanced lesions in vein grafts, like advanced coronary artery disease within a month or so. Next slide, please. Using this model, we performed the comprehensive and large proteomics analysis using two, strategies. The left hand side, I'm showing the static one-time point tissue, proteomics. On the right side, I'm showing the kinetic study, over time to compare the control IVC and vein graft lesion development over time. Next slide, please. From this analysis, we identified that PPA alpha is suppressed during disease progression. Please advance. Yeah. Yeah. Hit advance again. Yes. And one more time, please. One more time. Thank you. Our question was particularly, does PPA alpha have a causal role in vein graft disease development? Next slide, please. To answer this question, first we use the macrophage targeted lipid nanoparticle to deliver siRNA against PPA alpha. And as you can see here, with PPA alpha silencing, disease progression was accelerated. Next slide, please. Yeah. Oh, no. Please go back. Yes. Our question was then how we can do gain of function study. And then, fortunately, we had access to Pemafibrate, at that time, quite, new molecule. And we decided to use Pemafibrate for gain of function study. Please advance. Using Pemafibrate, we found that PPA alpha activation, decreased the lesion progression and macrophage activation as visualized by molecular imaging for MMP activity. Next slide, please. We also performed the metabolomics and lipidomics to understand that what's going on within the cells, are treated with PPA alpha. Hit advance, please. So we found that from a series of data that PPA alpha activation by Pemafibrate can mitigate hyperglycolytic state in LPS elastic macrophages, which is the typical sign of activated macrophages or so-called M1 macrophages. Next slide, please. So overall, we found that evidence from multiple assays that PPA alpha activation by Pemafibrate can reduce pro-inflammatory macrophage activation, partly through multiple changes in metabolic pathways. Next slide, please. So this is this result mirrors what we learned a lot in hepatocytes biology, and as you can see here, the comparison between glucose-rich state and glucose-poor state, and we believe that in macrophages, PPA alpha activation can trigger starvation signal to make the cell phenotype healthier. Next slide, please. Next. So this is the data from single analysis of macrophages treated Pemafibrate. I have no time to explain the data, but bottom line is we found that PPA alpha also regulates macrophage heterogeneity. Next slide, please. So AV fistula is also another major global burden. Obviously, this is important blood access in patients with end-stage renal disease. And then similar to vein graft, 50% of AV fistulas fail, in 1 year, and there are no medical therapies. Again, very similar to vein graft disease, due to high initial year event rates, the clinical trials for a new drug against this disease could be short and small. Next slide. As you know, the CKD has become major clinical burden, and this is the data in US. 80% of patients receiving hemodialysis have a cardiovascular disease, and therefore AV fistula failure is very important target for therapy. Next slide, please. We use the mouse model of AV fistula disease, in which we connect jugular vein to carotid artery. Next slide, please. Using this model, we also looked at the effect of Pemafibrate. So this is the ultrasound image of control, AV fistula in mice, untreated, in untreated animals. Next slide, please. So if you look at AV fistula of mice treated with Pemafibrate, you can see the more opening of the vein AV fistula, in these mice. And next slide, please. So this is a quantitative analysis. The bottom line is, Pemafibrate improves AV fistula patency in vivo. Next slide, please. So histology supported our finding too. More AV fistula were occluded in control animals as compared to Pemafibrate treated animals. Next slide, please. So this is the another publication from our group in JHA in last year. We demonstrated that Pemafibrate can reduce macrophage activation and lesion development in peak coronary arteries following stent implantation. We use intravascular in vivo molecular imaging in collaboration with Faruk Japa and Gary Tierney from Mass General Hospital. Next slide, please. So using this technology, we found that in a control peak coronary artery after stenting, you can see so much macrophage activity as visualized by molecular imaging agent to detect cathepsin activity. However, if you look at the Pemafibrate treated coronary artery, those signals were almost gone. Next slide, please. We measured the neointima volume and percent stenosis, and both parameters were improved by Pemafibrate treatment. Next slide, please. So we also found that Pemafibrate decreased cell proliferation and accumulation around stent struts in peak coronary arteries. Next slide, please. One potential mechanism was suggested by a series of in vitro culture experiments. And Pemafibrate restored the decreased differentiation of smooth muscle cells induced by PDHBB, and also Pemafibrate suppressed proliferation of smooth muscle cells. Next slide, please. So this is some unpublished data. We found that Pemafibrate can suppress macrophage activation induced by interferon gamma, as determined by decreased expression of TNF alpha and I1 beta, while conventional PPA alpha, Pemafibrate, and gemfibrozil cannot really do this job. So this is the major difference between Pemafibrate and conventional fibrates. Next slide, please. this data also compare the effects of pemafibrate and fenofibrate, suggest this data set indicate that the pemafibrate regulates cofactor recruitment to PPAR alpha at much lower concentrations as compared to fenofibrate. Next slide, please. This is another example of the effect of pemafibrate on mechanically injured arteries. As we saw in PIC study, pemafibrate suppressed the neointima formation after injury. But in this study, we compared pemafibrate and fenofibrate. As you can see in these graphs, pemafibrate decreased lesion development, but fenofibrate didn't cause these effects. Next slide, please. So from these results, I can say pemafibrate can attenuate lesion development of vein grafts in mice, AV fistula in mice, in-stent stenosis in PIC coronary arteries, and neointima formation in mouse femoral arteries. And the question here is that could vein graft failure, AV fistula failure, in-stent restenosis be clinical indications of pemafibrate? Next slide, please. These studies were done by very talented fellows and colleagues. And next slide, please. And wonderful collaborators. Thank you.